Today's lesson is going to be on prokaryotic gene regulation. We haven't talked about prokaryotes in a while, so I'm going to go over a few of their basic facts. Prokaryotic. Ooh, hello. Why does that pop up? There. Prokaryotic gene regulation. This is known as the Operon model. In this class, there are two different types of operons that you all need to know. We're going to start one of them today. The operon is found in prokaryotes only. So let's go over a few things that you may have forgotten about prokaryotes. Did they evolve before or after eukaryotes? Yeah, they're the first life forms on Earth. It is entirely possible that we may find alien life within your lifetime. That would change the science books forever. But if we do, it's not going to be some three-headed octopus monster. It's going to be a single-celled organism. Something that's going to be like, really? That's it? That's an alien? Yeah, it's life on a different planet. That's an alien. But again, it's very possibly going to be just single-celled organisms. Now, what would it tell you? Ooh, here's a good question. What would it mean if we go to Mars, like in the next 20 years, humans go to Mars, and they dig in the dirt, and they actually find microorganisms, and they have the same anatomy, structure, cell parts as the cells on Earth? What does that tell you?
Maybe that it's not random.
Okay, what else? Nikita?
Well, I don't know, common ancestry again, like
It could be common ancestry. We have a common ancestry, something on Mars. That would blow us away. All right, but we'll see if that ever happens. They're unicellular. An entire life form is just one cell. They belong to the domain Archaea or bacteria. Now, we have not done domains yet, and so it's understandable if you don't know it. But does anybody know what domain we belong to? No, that's the kingdom. That's one level below domain. We belong to the domain Eukarya. As humans, we are Eukarya, Animalia, Chordata, Mammalia, Primate, Hominidae, Homo sapien. That's us. They lack membrane-bound organelles. When you hear of organelles like a Golgi apparatus, an endoplasmic reticulum, mitochondria, chloroplast, hear me loud and clear. Those organelles are bigger than an entire prokaryotic cell. You don't have to draw this, but look. Let's say this is a bacteria that causes strep throat. That's a Streptococcus aureus bacteria. A Golgi apparatus is like this big compared to it. So it's impossible for these little prokaryotic cells to have organelles because the organelles are bigger than the cell. And they lack a nucleus. Now, when it comes to eukaryotes, we like to keep our DNA inside the nucleus. Prokaryotes don't have a nucleus. But do prokaryotes have DNA? Yes. Every life form on Earth has ATCG. So my question is, where is the DNA in a bacteria? Anyone know? Read?
nucleoid.
Very good. The nucleoid. In my experience, a lot of students have gotten the word nucleolus confused with nucleoid. Don't get those flip-flopped. The nucleoid is a region within the bacteria where the DNA can be found. A nucleolus is a structure inside the nucleus where ribosomal RNA is made. All right, and my last question, or my last point, What type of cell is more efficient? Eukaryotes or prokaryotes? What do you think? Reed? Yeah, by a mile. But Mr. Thorson, they're primitive. They're old. We're new. We're evolved. We're eukaryotes, baby. Yeah, we are. We are more sophisticated. We are more complex. It's complicated for us. Bacteria, it's easy. You ever heard the expression, they're so dumb they can't walk and chew gum at the same time? There's an old saying that, you know, back in your grandparents' day, they would say, they're so dumb they can't walk and chew gum. They're too stupid. Well, eukaryotes like us, we cannot walk and chew gum at the same time. We cannot do multiple things at the same time. For example, in 1961, researchers discovered the bacteria that's called E. coli, Escherichia coli. It's in your intestines, can regulate its own genes. Freddie, I want you to tell your pituitary gland to secrete growth hormone so you can get taller and more muscular right now. You can't do that. Why not? Because you cannot control the genes that control your pituitary. Bacteria can. Now, bacteria don't have a pituitary, but they can control their genes. Not only did these researchers discover that bacteria like E. coli can regulate their own genes, they can also do transcription and translation at the same time. We can't do that. They can do transcription and translation simultaneously. We have to do all of transcription first, and then we can go to translation when we're done. We can't do them at the same time. Imagine if you could come to play in high school and take AP Bio, AP Economics, AP Art History, Trigonometry, Lunch, Hope, and Spanish 3 all at the same time in 1 hour, and you're done for the day. You show up at 8:30, you're done at 9:30. That would be great. It's kind of tough. Well, if a prokaryote can actually learn, they probably could do that. They can do multiple things at once, which makes them more efficient despite the fact that we are more evolved. So these researchers they called this whole idea of bacteria's ability to regulate its own genes the Operon model. And this is what we're going to get into today. So what you guys need is, let's see, 1, 2, 3, 4, 5. You need about 6 colors and a highlighter if you have them. Because I really want our drawings today and tomorrow to really pop out to you and not just be some bland black, blue, or gray pencil or pen. So if you have multiple colors, get them out, get them out. All right. So there are several components to an operon, and this is how bacteria are able to regulate their genes. The first part that we're going to mention is the promoter. The promoter is a segment of DNA where RNA polymerase attaches to the operon. Now, guys, as a teacher, any teacher worth their salt can really have a sixth sense when their students understand something when they don't. And I'm getting bad vibes about the following question. Can anyone tell me what RNA polymerase does? Go ahead, Nikhil. From what? That's right. I want you all to check out Friday's lesson. See if this rings a bell. On Friday, we did transcription, and I broke it down to you in 3 stages. Stage 1, you have your DNA, but you only want to copy 1/2. What do you call the half that you do want to copy? The template. What do you call the half you don't want to copy? The non-template, okay? Next step, how do you actually get the DNA open so you can get to the nucleotides? Helicase. And then what enzyme is going to scan the template strand to make the mRNA? What enzyme would that be? RNA polymerase. So if RNA polymerase reads that DNA, and Lance, if RNA sees TAG, GC, TCT, what would the mRNA complement be for that? Last letter is T, so what's the opposite of T? A. If RNA polymerase sees TAG, GCT, CT, the opposite of that would be AUCC, GAGA. So in case you forgot, RNA polymerase creates mRNA nucleotides according to a DNA template. Are we clear on what RNA, do we have a better understanding of what RNA polymerase does now? If I ask you tomorrow, do you think you'd be able to tell me? We'll find out. All right, next one is an operator. An operator is a segment of DNA where a repressor binds to the operon. Now, I made the assumption that everyone knew what the word repress means, but that's not the case. So I want to tell you guys what the word repress means. I have dictionary.com open. There are 70,200,000 results for repress. Here's the basic definition. Subdue by force. Synonyms include suppress, quell, quash, subdue, put down, put an end to. Restrain, hold back, keep back, hold in, bite back, suppress. So let's say Adam is going to get a fight with Lance because Lance said you're a doo-doo head. You're not, okay? You don't need to cry. Yeah. And so I, the teacher, see these two students about to fight. What does the teacher normally do? They step in between them. So I'm going to just hold him down, and I'm just going to lightly squeeze his shoulder. And he goes, "Oh, my shoulder, Mr. Carson, you're so strong." Right now, I am repressing him. As the repressor, am I active or am I inactive? I am active. And since I am active, is he on fighting mode or is he off? Non-fighting mode. He's off. As long as I'm here repressing him, he's off. But then I release. Am I active or inactive? I am inactive. Is he on or off? He's on. So what I need you to make a connection with is that when a repressor is active, whatever it attaches to is going to be shut down. It will be turned off. And when a repressor is inactive, whatever it attaches to is turned on. So I'm going to write that down. Remember that to repress means to subdue, to stop, to quell, to put down. What is it? To just subdue by force. So when we have repressors, an inactive repressor it will result in a Operon is on. And an active repressor means the Operon is off. Okay? All right, next, we have structural genes. Structural genes are several genes that code for I don't know what that word primary. That code for the needed enzymes. And go way back to unit 1, guys. What type of molecule is an enzyme? What type of macromolecule is it? Right, it's a protein. The structural genes are what are transcribed. And the last one we have for today is the regulator gene. The regulator gene is not part of the operon. It is controlled by the promoter. And it manages the repressor. Okay. And that concludes your cast of characters for an operon. What we're going to do now, once you all are all caught up, is we're going to draw them and identify where they're located within an operon. Okay, so the first thing that I'm going to draw, and this is all going to look like DNA, so we're going to have the twisty helices. The first part we're going to draw is way over here on the left. We're going to draw the structural genes. So I'm going to keep everything color coordinated. Or did I say structural genes? My bad. Regulator gene. I keep getting those crisscrossed. Okay. Keanu, I want you to use your big boy voice. What does this say?
It's not part of the person, no, the Operon.
Well, kind of terrified. The regulator gene is not part of the Operon. So here's what I'm going to do, guys. I'm going to simply draw a bunch of garbage, trash, space filler DNA to simply show you that the Operon and the regulator are far apart. So don't write this, just listen up. I am going to draw the Operon way over here somewhere. I'm going to keep the regulator far on the left. So what I'm going to do is I'm just going to draw some DNA to just fill this void between the regulator and the rest of the Operon. So this blue DNA I'm drawing is absolute nothing. It's just a space filler. That's all. So you don't need to label it, just it's a space filler. What I'm doing is I'm trying to demonstrate to you that the regulator, by definition, is not part of the Operon. Everything else that I'm going to put over here on the right, that will be the Operon. So now let's get back to it. The red is going to be the promoter. The blue, the navy blue, is going to be the operator. And finally, the green is going to be the structural genes. And everyone, did I say everyone? Everyone, the promoter through the structural genes are the Operon. All right? All right. That is the basics. We haven't gotten into the two different types of operons that you need to know. I'm simply just drawing you the baseline. The last thing I want to add actually is something very simple. I'm just going to draw a little repressor. That's all. Nothing major. It will play a role later. You'll see. By the way, is that repressor attached to the Operon right now? So would we say the repressor is active or inactive? It is inactive. Is the Operon on or off? Good. 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 Okay. So now we can go ahead and start sneaking into Thursday's lesson. And let's talk about the first Operon that you need to learn about. This is called the trp Operon. Do you recall what amino acid is abbreviated trp? Tryptophan is correct. Tryptophan is an amino acid. For humans, it is an essential amino acid. For bacteria, it's non-essential, which means they are capable of making it. A good portion of your mastering homework this weekend is based on the trp Operon and the lactose Operon. We'll get to the lactose tomorrow. So I want you guys to, once you're all caught up, I want you to stop writing. I want you to really listen to what I had to tell you, because a lot of this just, you need a second to think it out. Really, you have to connect the dots. So, Freddie, do you eat lunch here at Plain every day? You do. Do you make your own, or do you bring your lunch, or do you buy it? You bring it. Who makes your lunch?
Me and my mom.
Oh. So, let me ask you this. Do you bother to make your lunch if your mom has already made it for you? Right? Makes no sense. So what I want you guys to understand the logic here is there's no purpose, there's no point in making something if you already have it. Chloe really wants that jacket from Nordstrom. She's been eyeing it. She's like, oh my gosh, it's so beautiful. I want it. I'm going to save up for it. Nordstrom's not cheap. She goes home. It's on her bed. Her mom bought it for her as a gift. Hey. Does she need to go to the store and buy it now? No, there's no point. In the 1980s, there were wrestlers that were known to have taken testosterone shots, steroids, right in the butt. Is their body going to bother to make testosterone if they're getting it from another source? No, they're not. So the point here I'm trying to make is, in a cell, there's no purpose in making something if the something is already available. I don't need to go to Publix to get a pub sub if I have a pub sub in my fridge. It's there. Why go out and get another? So, if you can understand that logic, you'll understand this operon. The trp Operon produces tryptophan when tryptophan is not present in the cell. The tryptophan operon produces tryptophan when there is no tryptophan. So the trp Operon has two basic switches. It's on or it's off. When it is on, I want to see if you guys might be able to tell me. Listen, it makes tryptophan. Ask yourself, why would it have to make tryptophan? Is there tryptophan present or is there not tryptophan present? Good. So under what conditions would the tryptophan operon have to be switched on? Grace Mary?
When no tryptophan is present.
Tryptophan is absent or not present, however you want to say it. So the tryptophan operon is on when there is no tryptophan present. When there's no tryptophan present, it needs to be made. Okay, so in that case, under what conditions will the tryptophan operon be turned off? Anyone? Nikhil? That's it. Nikhil, do you, when you're in college and you have to go fend for yourself and get food for yourself, do you need to go out and spend food on groceries if your parents just came to visit you and took you to the grocery store? You guys are going to love that, by the way. When you're in college, your parents come to visit you and you're like, hey, Mom, can we go to Publix, please? And you just load up on food. It's going to be great. Publix is going to be, going to the grocery store with your parents is going to be one of your favorite things when you're in college. All right, so it's off when there is tryptophan present. Why make something if you already have it? Stop making it. All right, so what I'm going to do for you guys now is I'm going to draw what happens when the tryptophan operon is on. And that's we have 6 minutes, so this is all I really have time for. So let's go ahead and draw everything, but let's take it a little easier on ourselves. Rather than draw the twisty, 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 I'm just going to draw lines with the representative colors to represent the structural, regulatory, promoter, operator, all this stuff. All right. So I use gray for the structural. Actually, hold on. Or the regulator, I mean. I keep getting those words confused. Then I just have that gap. Then we have the promoter. Then we have the operator. Oop, I got to do something right on that. Hey guys.
Teachers, please.
I did this on purpose. Draw that, include that.
Tryouts continue today and tomorrow at 7 p.m. Remember to have your athletic clearance to be able to participate. Flag football conditioning also continues today at 3:45 p.m. on the football field. Anyone who wants to participate must have an approved athletic packet on file with the school before you can come out. Please see Coach Conte in room 12 if you have any questions. Softball tryouts are continuing through Thursday, January 28th at 5:30 at the softball field. Please make sure your athletic packet is turned in and approved. Seniors, you're now dismissed.
Okay, I'll let you guys get caught up. We should be able to finish this by the bell. Okay. For the third time today, see if third time's a charm. What is the name of the enzyme that will read a DNA template and produce mRNA? Love it. Oh, I'm so happy. Yes. So this is RNA polymerase. I want you to look back on your notes on promoter, operator, structural, regulatory. Tell me, what part of the Operon does the RNA polymerase attach to? What do your notes say from just 20 minutes ago? Rosemary?
The promoter.
That is correct. So if you guys want to give your writing hand a little breather, you probably aren't going to be able to do this on your paper. If you have a tablet, you can just simulate it like I'm about to. Here we go. This is the RNA polymerase. It is going to attach to the promoter right there. And now here's the moment of truth. Listen up. I left that little notch right there in the operator. Do you see a repressor plugged into that? There is no repressor plugged into it. So, is there any obstacle preventing the RNA polymerase from scanning the DNA? There's nothing standing in its way. So the RNA polymerase is going to move down and it's going to scan the structural genes. Now, what do you get when RNA polymerase has scanned a DNA template? What do you produce? mRNA is correct, and here it is. And once mRNA has been formed, what's the next step? What does mRNA go through? It goes through translation. What is produced in translation? Proteins, that's right. Proteins are very weird, odd-shaped squigglies. These are going to be enzymes that can produce ATP. Or not ATP, wow. TRP, tryptophan. So the point here, guys, is that there was no tryptophan present in the cell. The cell is going to make its own. That is what is happening here. One last thing, and I promise this is the last thing I'm going to draw. What shape would you say the little notch is like? Is it rounded or square shaped? Square. Let's take a look at the repressor. Can this little knob right there fit into that operator? No. We're going to find out how that changes tomorrow. This right now is an inactive repressor. And that is it for today. We will continue tomorrow.